GMO is also known as genetically modified organisms. Genetically modified organisms are those organisms whose genomes have been altered using genetic engineering technology. GMO usually is when genes from the DNA of one species are extracted and artificially forced into the genes of an unrelated plant or animal. These organisms are also called transgenic organisms due to the alteration of their genome. GMO was first identified in bacteria because of how abundant, cheap, ethical the bacteria was to researchers. Researchers used plasmid, which is a small circular self-replicating piece of DNA that can be readily isolated from bacterial cells and could be altered by inserting or deleting specific sequences of DNA. In order to change the genetic genome of the bacteria, researchers used restriction enzymes, which are proteins that cut DNA at specific sequences. Some restriction enzymes generate blunt ends, cutting across both strands of DNA, while others generate a staggered cut, producing sticky ends. After the cut, the same complementary bases will be inserted with the target gene or the other DNA into the cell so that the nucleotides match with their complementary bases and with the edited gene inside the cell's DNA. Adenine will go with thymine and cytosine with guanine. For bacteria, their plasmid genes will be edited and inserted into the cytoplasm, but for eukaryotic cells, researchers will use the genetically modified gene and place it into an egg of an organism. Then they would insert the egg into a surrogate animal to give birth to the transgenic organism. The new edited DNA is also called recombinant DNA. GMO can help thousands of people with diseases, famine, and genetic disorders. However, there are many controversies as to whether GMO on humans is ethical or healthy in the long run. In a recent study, researchers have found a new discovery producing the mechanical properties of composite silk in heritable transgenic lineages. Silk has been produced for thousands of years primarily because it displayed high tensile strength and extensibility, making it tougher than any man-made synthetic material. Researchers wanted to find out what makes silk from spiders so naturally durable and strong. Using different lengths of MASP1 derived from the dragline silk protein L. hesperus, researchers wanted to analyze this gene expression on heterozygous and homozygous genotypes of the edited gene. They first implemented multiple genes, such as DS-RED, into the transgenic lineages to have physical evidence and identification of which organisms had been genetically modified. This is called genetic marker. The markers genes for these moths were to have glowing eyes and red fluorescent light. They first wanted to see if heterozygous or homozygous genotypes had anything to do with the quality production of silk. Then they wanted to see if the length of the MASP1 would affect the quality of the production of silk. They generated 5 generations of transgenic moths with the MASP1 gene and analyzed generation 4, which was the heterozygous group, and 5th generation, which had the homozygous group. After analyzing the cocoon of the 4th and 5th generation, researchers found out that there was no significant difference between the 2 generations of silk production. They also discovered that the silk from the cocoon of the 16 times MASP1 had twice as strong silk than the original organism. This ultimately proved that the length of the repeating motif protein MASP1 is what truly determines the silk's toughness and elasticity.